Hi students, welcome back to our YouTube channel. Today is our fourth lecture on carbohydrates topic. In this lecture, we are going to discuss the reactions of monosaccharides. Okay, so first of all, the first reaction we are going to discuss, that is tautomerization or enolization. Okay, like as you know that the tautomerization is defined as the process of shifting a hydrogen atom from one carbon to another. Okay, so that will produce anidiol. Okay, so that process is known as tautomerization. Like in general word, for example, here we have the two carbons and the shifting of the hydrogen from one carbon to the other carbon, that process is known as tautomerization. Okay, so remember here the sugar processing anomeric carbon okay, carbon atom that undergoes tautomerization in alkaline solution. So here it should be alkaline solution. Okay, how is it possible? Like when glucose is kept in alkaline solution for several hours, like there is a glucose that is kept in like alkaline solution in several hours, so what will happen? It undergoes isomerization. To form like D-fructose and like the next one, that is D-mannose. Okay, so this reaction is also known as Lobreedy-Buran-von Eckenstein transformation. Okay, how is it possible? I am going to show you the three forms of like the three different structures. The first one is D glucose, the other one like D fructose, and the third one that is D mannose. Okay, now the first one, in the first structure, okay, here you can see the aldehyde group, okay, So that is the first structure for the glucose. Okay, so remember R is actually corresponds to the ends of 3 carbons. Okay, so for the D fructose, that contain ketonic group, so I am going to make it. Okay, like the other 3 carbons are represented by R. Okay, in case of D mannose, so that is a structure. Okay, and remember R is actually R is equal to 3 carbons. Okay, so that is in the common structure in all these 3 structures. Okay, let me, now you can see. Now in how the anidiol formation take place by the process of tautomerization, so I will show you here. Okay, in the first case, the shifting of hydrogen to the next carbon. Okay, so here you can see it will form anidiol formation, so that is COH H. Now you can see here, like there is OH, but in this case when the hydrogen is shifted to the next carbon, so it means there is a double bond in between carbon. Okay, and the next one there is hydrogen and OH. Okay, so same happening here in the D fructose case, that will make a same anidiol formation, and same happening in the D mannose structure. Okay, so here I am going to show you all these 3 transformations, like D glucose, D fructose and D mannose, that can be converted into anidiol. Okay, so that is common, so it means here the name will be anidiol formation. Okay, and remember anidiols are highly reactive. Okay, so hence sugar in alkaline solutions are powerful reducing agent. Okay, in the alkaline solution. So that is over the first property or the chemical property of the monosaccharide. That process is known as like the tautomerization or annualization formation. Okay, so now we are going to discuss the next one reaction, that is reducing properties. Reducing properties. Okay, as I already discussed that the sugars in alkaline solutions are powerful reducing agent, so it means the sugar are classified as reducing and non-reducing. So sugar can be classified as reducing sugar and non-reducing sugar. Okay, so the reducing sugar Remember, the reducing property is attributed to the free aldehydic or keto group of anomeric carbon. So it means here that is attributed to free aldehydic or keto groups of anomeric carbon. Okay, I uploaded a detailed video on anomeric carbons, so you have to search in our the contents. Okay, now in the laboratory many tests there are the different types of the labs test for the monosaccharides to identifying the reducing sugars or the reducing actions of the sugar. So these include Benedict tests, filling test, the next one is Burford test, okay, and many more. So remember, the reduction is much more efficient in the alkaline medium, as we already discussed, than in the acidic medium. So you have to remember, the reduction is more favorable in alkaline medium. Okay, rather in acidic medium. So remember, the anhydrol formation or anhydrol forms here we did in the previous property that the anhydrol or sugar reduce cupric ions of copper sulfide to cuprous ions, okay, which form a yellow precipitate of cuprous hydroxide. For example, here is copper sulfate that will convert it into copper plus 2 ions, okay, and when that ions are reacted with the anhydrols, so that will convert it copper plus 2 to copper plus 1, like reduces, okay, and that will make, along with the water molecule, like here, that will make copper hydroxide, okay, and that copper hydroxide can be converted into copper oxide plus water molecules, okay. So here you can see the conversion of the copper plus 2 to plus 1, and then that can be formed copper oxide, or instead of copper oxide, first it will form like cuprous hydroxide and then a red precipitate of copper oxide. So cuprous hydroxide color will be yellow and the copper oxide color will be red, okay. So it may be noted that the reducing property of the sugar cannot help for the specific identification of one sugar, so that is like a general reaction, okay. So you have to remember the next the second characteristic, or you can say chemical property of the monosaccharides, that is the reducing property, okay. Now we are going to discuss the third one, that is oxidation. oxidation is, as you know, depending on the oxidizing agent used. Oxidizing agent, like oxidizing agent that oxidize the other one, but reduces itself. Remember it, okay? So here is, depending on the oxidizing agent used, the terminal aldehyde or ketone or the terminal alcohol of both groups may be oxidized in the monosaccharides. Like the first one, considered like a glucose, oxidation of aldehyde group, that will make like the glucanoic acid. Here you can see the appearance of the carboxylic acid. So result in the formation of the glucose that will be converted into glucanoic acid. Okay, and the second one, that is like the first one is in case of aldehyde. Here you can see. Okay, now the second one, the oxidation of like the terminal alcohol, that is that can be again converted into like carboxylic acid. So here that will lead to the production of glucuronic acid. Okay, so that is the in the oxidation, like that is the oxidation property. Okay, now the fourth one that we already discussed, like reducing properties. So here we will discuss in detail about the reduction. Okay, so reduction is like in the process the use of reducing agent. Like the reducing agent is the one that reduces the other one but oxidized itself. Okay, so here like such as we can use here the sodium amalgam as a reducing agent. Okay, the aldehyde or keto groups of the monosaccharides is reduced to corresponding alcohol. Remember, aldehyde or keto groups that will reduce to form corresponding alcohols. Okay, let's suppose here the general formula is like here you can see aldehyde group that will be reduces to form corresponding alcohol. Like for example here we can add the 2 hydrogen. So then here you can see these 2 hydrogens. So the important monosaccharides and their corresponding alcohols I am going to show you. For example, D-glucose, that can be converted into D-sorbitols, okay, and like the same, like D-mannose, that can be converted into D-mannitol. So you can see like the indication of ol, that is indicated there is that is the alcohol. And same here, like the third one, there are a lot, but here like the D-ribose, that can be converted into D-ribitol, okay. And remember, like sorbitols and like delketols, when accumulated in tissues in large amount, cause strong osmotic effect leading to swelling of cells, okay, and certain pathological conditions like cataracts, okay. And there are also like the other condition that is known as nephropathy, while the mannitol, that is useful to reducing interacranial tensions of forced diuresis, okay. You have to remember that. And now the next property, that is dehydration. Okay, so dehydration is, as you know, that the well-known dehydrating agent that is commonly used, that is the sulfuric acid. We already did the video of sulfuric acid treated with the sugar to make like a snake effect. Okay, so you can search that video in our like the contact sections. So same thing happening here, like when treated with the concentrated sulfuric acid, monosaccharides undergo dehydration with the elimination of 3 water molecules. So thus the hexoses give hydroxymethyl, while the pentoses give like furfuryls on dehydration, okay. So for example, here is the glucose, D-glucose, in the presence of H2SO4, there is the elimination of 3 water molecules. And now the next one, that is like 1 2 3 4 5 and 6, okay. So here that is the product form, okay, and that product name is hydroxymethyl furfuryl, okay. And remember, the furfuryls can condense with the phenolic or phenolic compounds, like for example we can use like alpha naphthol to form colored products, okay. So this is the chemical basis of popular, that name is Biles Test, like colored compounds or colored product formations by the use of alpha naphthalene okay, that process is known as Biles tests. Okay, so in case of oligo and polysaccharides, we discuss later, we will do in like the later lectures. Oligo and polysaccharides, they are first hydrolyzed to form monosaccharides, and like they are first hydrolyzed to form monosaccharides by acids, and then it is followed by the dehydration. And in the dehydration we will do like the next test, that is known as Biles test. In the Biles test, pentoses react with the strong acids like sulfuric acid, okay, to form few here furfuryls derivatives. Okay, and which is turn react with the orchinols. If we react those derivatives with orchinols, then that will make a complex green colored complexes. Okay, so Biles test is used for the detection of xylose, okay, in urine, okay, so remember that. Now the next test, okay, now the next property we are going to discuss, that is the Osa Zone Formation. Okay, so Osa Zone Formation, here is we can use a phenylhydrazine, okay, that is like phenylhydrazine in acetic acid, that is present in like the solvent or the solution that we can use like the acetic acid. So phenylhydrazine in acetic acid, when boiled with the reducing sugar, okay, so it will form Osa Zone Formation. Okay, remember it. So Osa Zone Formation, for example, we are going to take the glucose, and I am going to give like the R for the other groups, react with the phenylhydrazine, okay, so that will make Okay, first the intermediate, that is glucohydrazone. Okay, that intermediate is known as glucohydrazone. And then there is like the addition of more phenylhydrazine, like here phenylhydrazine we can write like the phenyl group C6H5. Okay, and same happening here. Okay, so okay, that will make the first group is same, while the next one there is again the addition of phenylhydrazine. Okay, and that product is known as gluco Osa Zone Formation. Okay, so R that can be represented like C3 to C6. So remember, the sugar that differ in the configuration on these two carbons, like the two carbons are C1 and C2 carbon here, gives the same of the Osa zone, since the difference is masked by the binding of phenylhydrazine. Okay, so thus glucose and fructose or mannose gives the same type like needle shape Osa zones. So that will make needle shape Osa zone formations. Okay, so reducing disaccharides also gives Osa zones. Disaccharides also gives Osa zones. Okay, but like as the monosaccharides give like a needle shape Osa zones, but in the disaccharides, like for example maltose, that will give a sunflower shape. Okay, while the lactose that will give like powdered puff shape. Okay, so that is in the Osa zone. Now the next one, the last one, property of the monosaccharides, or the reaction of the monosaccharides. Okay, it's not 4, it's I think 7. Okay, that is the formation of esters. Okay, in the formation of esters, the alcoholic group of the monosaccharides okay of the monosaccharides that may be esterified by non-enzymatic or enzymatic reactions. Okay. Like esterification of carbohydrate with phosphoric acid, esterification with phosphoric acid, okay, like esterifications of carbohydrates. Okay, so is a common reaction in the metabolism. Okay, so like glucose 6 phosphate and glucose 1 phosphate are the good examples in which like the ATP donates the phosphate moiety in the ester formations. So if you don't know about the ester formations, so we will make a detailed lecture on the esterification process in the next videos. Okay, so that is the last property of the monosaccharides reaction or the monosaccharide the chemical property. So in the next video we will discuss the glycosides, and then we will do like the polysaccharides and also like the disaccharides and oligosaccharides. So if you like our video, then kindly subscribe us and share our channel. Thank you so much. We'll see you in the next video. Goodbye.